with today's lesson, we enter the second part of the course. First part was about epistemology. Now we talk about the basic instruments that the scientist uses to do his research. So specifically we talk about isolation of the variable of interest and positive and negative control. What's your experience with these two topics? Have you heard of positive and negative controls? Yes, you have. Less likely you have heard about isolation of the variable of interest. You confirm. Right. So, let's see what's all about. Okay, rather than giving you a formal theoretical introduction to these concepts, I'd like to start with a specific paper, a very famous one by the way, which we'll use in this lesson and in several others to talk about different topics, providing right away examples that illustrate what... Non accessorio. Sì, l'ho fatto. So these topics will be illustrated by the examples we are going to go through. So let's see what the paper is. The title is Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. You probably don't recognize this paper. Perhaps you recognize the name of the last author, if you can read it. Yamanaka. You heard about him? Okay. In 2006, I think. Yeah, it should be here. Yes, 2006. He discovered a way to convert somatic cells, that is cells taken from any part of the body of an adult animal, an adult mouse in this case, to convert them into pluripotent embryo-like stem cells. Now that was a major breakthrough, and in fact 5 years later he was awarded the Nobel Prize for this. I remember when I first saw this paper, it was, I have a precise memory of it. It was a Saturday morning, I was alone in the lab, I read this paper and I said, no, it's impossible. And so, and then I went through it, and I was enthusiastic about the paper and about the approach, which is kind of surprising, but it serves well the purpose of illustrating today's topics. Okay, so what does the title mean? What I've just said, that Yamanaka found a way to convert fibroblasts, in this case, but this is a very general method, it applies to a wide variety of cells, into pluripotent stem cells. And did so by using defined factors. Now, to better appreciate this paper, I'll provide you with some background. That is what Yamanaka knew at the time, which constitutes, yeah.
Salve, troppo, scusi per il ritardo.
As I was saying, what Yamanaka knew at the time and that constituted the background of the study he undertook. Of course, this paper, this study, like every other study, does not come out of a vacuum. It stems from previous knowledge. What was the relevant previous knowledge? Let's see. First of all, this paper here, in 1988, described a new way, a method to keep in culture stem cell lines derived from human blastocysts. So, for the first time, we could keep in culture human embryo stem cells in a defined way. Now, that gave the opportunity to study very early embryo development, as well as try to use those cells for studies in development and eventually even in regenerative medicine. But it was ethically controversial in that some people think that it is not ethically acceptable to destroy human blastocysts to do research. So, although these methods became available, it was not widely used. Also, the starting material was not widely available because, of course, human blastocysts, the blastocysts people were using were basically the leftovers of in vitro fertilization attempts. The extra embryos that are often not used because some others before them take in the uterus of the mother, and so there is no longer need for the remaining embryos that have been generated in the procedure. So the next paper, in fact, precedes this one by quite some time, because in 1962, in the early 60s anyway, John Gardner published a series of papers showing that it was possible to generate an adult frog by doing this amazing thing. Taken a somatic cell, in this particular case an intestine cell, take out its nucleus and transplant it into an egg that had been deprived of its nucleus beforehand. So the egg is now without a nucleus, and you transplant into it a nucleus isolated from a somatic stem cell, a somatic cell. Surprisingly, this reconstituted egg is capable of developing into an embryo, and at times, infrequently, but measurably, it even develops into an adult frog. Now, these experiments were very important at the time because for the first time gave an answer to a major question that there was in biology and in molecular biology. What is differentiation? There were at least two schools of thought about differentiation. One argued that differentiation was due to selective loss of genes not needed in a specific lineage the cell was differentiating into. Another school supposed that instead it was simply differential gene expression that regulated differentiation, and that there was no loss of genetic information. Now, of course, we know that with very minor exceptions, lymphocytes, the second theory is correct. But at the time, showing that you could take a somatic cell which supposedly might have lost parts of the genome and recover a whole animal clearly demonstrated that this cell had not lost anything. So that was important, and in fact Gardner got his Nobel Prize together with Yamanaka, again to confirm that these studies were provided crucial background for what Yamanaka did later. Next, for a long time it was possible to do this nuclear transplantation into eggs only with frogs. It was not possible to do it with other animals. It was not possible specifically to do it with mammals. So people thought, well, perhaps frogs are somehow diverse. In mammals it will never work. There are additional layers of regulations. We are different regulation. We are different from amphibians. But then, in 1996, Ian Wilmut published the famous Dolly experiment. This is Dolly. Actually, this is Dolly. That's his mother, her mother. The experiment was just like that of Gordon. Wilmut took an oocyte from the mother and transplanted into it the nucleus coming from a mammary cell, in that case. Any somatic cell. The oocyte now gave rise to an embryo, which gave rise to an adult sheep. So it was clear that nuclear transplantation worked with mammals as well. Since then, it's been done with a variety of species, of mammalian species and others, and so it's in some laboratories it's nearly routine, but it's lost importance because we now have more advanced techniques to do similar things. Another piece of background information that was critical for Yamanaka's thinking was the ability to, again, convert somatic cells into embryo-like ones by an entirely different means, which was cell fusion. Cell fusion was a very important technique back in the 70s and the 80s of the last century. And it was shown, how does it work? In the presence of specific substances, either chemical or viral, cells that come in contact with one another fuse their membranes, much like this, and become a single cell called a heterokaryon. A heterokaryon is the hybrid cell before it divides for the first time. It is called heterokaryon because it has two separate different nuclei, each coming from one of the progenitor cells. And once it undergoes the first mitosis, of course, nuclear membranes dissolve, and so they, after mitosis, the cells have a single nucleus, which is the sum of the two, but they are no longer binucleated like these. Okay, so if people fused a somatic cell with an embryo stem cell, the uniform result of the experiment was that the nucleus of the somatic cell acquired characteristics of the embryo. That is, it began to express embryo, typically embryo genes, and behaved like an embryo nucleus in all respects. So there was something that was capable of acting in trans from one nucleus and convert the other into a copy of itself. So both nuclear transplantation and cell fusion were techniques, sort of holistic techniques, that in some magical ways were able to modify the destiny of nuclei or even of cells of whole animals. But they were not defined. Nuclear transplantation was transplanting a whole nucleus into different cytoplasm. Fusion was putting together two nuclei in a common environment. We didn't know what we were doing here. We just knew the outcome, that was this. So a step forward was another major paper that came out in 1987, which showed for the first time that it was possible to convert a cell type into another by transfecting a single gene called MyeloD. Specifically, it was possible to convert fibroblasts into myoblasts by introducing MyoD into the fibroblasts. This was a major step forward because for the first time we had a genetically defined way to convert a cell type into another. It works with other cell types, but it's not as universal as the Yamanaka results. Anyway, the converted myoblasts acquired all characteristics of natural myoblasts, including, of course, the ability to differentiate terminally into muscle myotubes. So this was the background. And then came Yamanaka with his amazing paper, which when I read it for the first time, I said, if it were a proposal for a grant application, if Yamanaka were to ask money for this experiment, nobody would have granted that money because what he did was sounded crazy, but it worked. And in science, remember, it doesn't matter how crazy is the starting point. Something that works, something that teaches you something, something that answers your question is always right. Even if the premises were wrong. Never mind, because all we strive for is understanding. If in the end your experiment lets you understand something, it is successful. So, what did Yamanaka do? Armed with the background I just presented to you, he engineered cells, mouse embryo fibroblasts, with this construct. A gene, beta-GIO, which expressed a hybrid molecule, never mind. The GIO part is encodes resistance to an antibiotic called G418, it should be here somewhere, here, G418, which normally kills eukaryotic cells. It's not an antibiotic, it works on both bacteria and eukaryotic cells. It's a poison. And so he put this gene under the control of the FBX15 locus, of the FBX15 promoter, which is only active in embryo stem cells. So his reasoning was, if I'm able to convert these fibroblasts, first of all, the fibroblasts should not express beta-GIO when they are fibroblasts, because the promoter doesn't work in fibroblasts. It is not active. If I succeed in converting these fibroblasts into embryo stem cells, then the promoter will activate, will start transcribing the gene, and the cells will become resistant to G418. They will survive in the presence of G418. And this is a pretty standard selection technique, and in one of the future lessons we'll talk more about this. And then he basically screened the literature, and as he told the story later on, he interrogated many of his colleagues. Of course, he had a strong background in stem cells, in natural stem cells. He scored literature and eventually came out with a list of 24 transcription factors that, in his opinion, were potentially capable, some of them at least, of converting a somatic cell into an embryo. And then he did this apparently simple but also crazy experiment. He devised 24 retroviruses in order to efficiently transduce these transcription factors. 24 recombinant retroviruses, each one of them expressing one of the 24 transcription factors. And he infected the cells with all these viruses together, hoping that somehow some of the cells would convert into embryo cells. And this is the very simple first experiment that it shows in the paper. We have a plate. This is a tissue culture dish, a petri dish, which is photographed from above. And the black dots that you see here are cell colonies. When a cell grows in an empty space, forms a colony because the cells move, spread, and form a circular colony. Okay, this is the plate in which the factors have been transduced into the cells. And this is a mock plate. What is a mock plate? Mock means, in Italian, means finto o presa in giro. So here he used an empty retroviral vector. So he pretended to transfuse the cells but didn't do it. And then subjected the cells to challenge with G418. Both plates were added with G418, and here no cells survived. Here a few colonies formed, implying that some cells had been converted into embryo stem cells. Now, this is not a proof yet, because you might argue that, well, perhaps the promoter has been activated by something that happened in a few cells. Keep in mind that probably in the beginning there were hundreds of thousands of cells per dish. So you might imagine that perhaps a mutation occurring in a few of these cells might have activated the promoter. But in fact, this mutation did not occur in the mock dish. So it would be a strange type of mutation. But it's not inconceivable that this is something other than conversion into embryo stem cells. For the time being, all we know, all Yamanaka knew, is that some colonies formed. Then he picked those colonies, took the cells from the dishes and put them in cultures, one colony separated from the others, and showed that while mouse embryo fibroblasts here. Oh, by the way, these are called mouse embryo fibroblasts because they come from a mouse embryo. But it has nothing to do with embryo stem cells. It is just a practical thing. Fibroblasts coming from an adult would grow only for a short time in culture, so it is more convenient to get them from an embryo. But they are fully somatic cells. Okay, they grow in this way. They're flat, they're scarcely visible in this photograph. And here is how these cells, these colonies, grow in culture. You see, they form bumps, they form blobs of cells, scarcely attached to the bottom of the plate, and they look very much like the way natural embryo stem cells grow. So, not only they were G418 resistant, they also were grew similar to natural embryo stem cells. That was very encouraging. Now, why? This is a question for you, so start thinking. Why did Yamanaka bother to have a second dish? Why wasn't this dish enough for the experiment? Right?
Because you need to have the proof that what you're looking for wouldn't occur naturally.
Yes, okay. Anything to add? No, okay. So that's the correct answer. It is something I was alluding to before. That is, how do you know that this conversion, or this whatever it is, doesn't happen naturally, spontaneously? You know it because you have a control dish. What is a control dish? It's a dish which has been treated exactly like the test one, this is the test dish, except for one variable. This is what we call the independent variable. Independent means you decide where to put the variable, in what amount, for how long, whatever. It's under your control. Every other variable in the dishes, thousands, millions of variables, you might imagine of looking for anything in these dishes. Expression of whatever gene you're interested in, presence of proteins, speed of replication, G418 resistance, resistance to radiation, whatever. Everything else should be the same in the two dishes. Whatever you find in this dish then must be causally linked to the independent variable, because that was the only difference in the beginning of the experiment. So whatever you observe at the end of the experiment must derive from the independent variable. Is that clear? Okay, so this is crucial in an experiment. And by the way, as we'll discuss in a future lesson, it distinguishes experiments from observations. In observations, you cannot isolate a single variable, because observations are made on natural objects, which are naturally different from one another. So you cannot isolate a variable in the sense of having all other variables identical in all subjects of your observations. In an experiment, at least in principle, you can do it because you create these dishes and you make sure that they are identical and that they are treated identically and that they are in the same environment and that they are manipulated together in order to make them indistinguishable from one another, from each other than for the independent variable that you intentionally put in one of them and not in the other. So experiments, at least from the logical point of view, ensure that there is a causal relationship between the independent variable and the outcome of the experiment itself. So now there is another question for you. What is the independent variable in this experiment? What's different between the two dishes? Wait a second. Give them time to answer.
The resistance to the antibiotic.
That's the outcome of the experiment.
Okay.
Some cells have become resistant, but that's the end of the experiment.
Okay.
In the beginning, what was the difference between the two dishes?
The virus.
The viruses, yes, that is in fact it was the transcription factors transduced by the viruses, right. There is a problem though. I said Yamanaka put into this dish 24 viruses, which seems to go counter the idea of having one independent variable. How do we reconcile that?
The presence of the beta-gene, the expression of beta-gene gene, because it becomes the variable.
No.
No.
No, that's again the outcome. The expression of the gene is the end of the experiments. It happens temporarily in the end of the experiment, or at least during the experiment. It's not there in the beginning. In the beginning, neither cell dish expresses beta-gene. So the question is, I've said we had to isolate one variable, but Yamanaka put 24 of them here. So was that not a proper experiment? Is Yamanaka a poor scientist?
You wanna know,
But probably not.
Maybe the important part is that in the mock one there is no virus.
In fact, there was a virus. There was an empty virus,
Empty virus.
identical to the viruses used here. But still, we haven't identified the variable that distinguishes the two dishes in the beginning of the experiment. It's easy. It's basically dialectic, the answer. Can we not say that we have a collective variable here? That the variable is all 24 transcription factors together. You have thought about that, but you didn't dare voice your thinking.
I wasn't sure.
You don't have to be sure. In science, oftentimes, most of the times, you are not sure what you're saying. Okay? So, say what you're thinking. The way we discuss things in science is you say one thing. I say, well, your thinking is interesting, but you didn't think of that. And she says, yes, we might say this. And he says, we might modify it this other way, and so on. So we build a proper model, a proper approach to the problem, bit by bit. None of us really conceive an experiment altogether at one time. So we have to voice our opinions, otherwise we don't build anything. Okay? From now on, please say whatever you think, even if you're not sure it is correct. Alright, so the difference between the two dishes was the presence of the 24 transcription factors at the same time. Good. So we can be sure that the 24 transcription factors altogether, as a whole, were responsible for the eventual resistance of the cells to G418 and their peculiar way to grow. Good. Now, let's stick for the moment to this point. Isolation of an independent variable. Admittedly, this was a tricky point in this experiment because, as I said, this was a collective variable. But conceptually, you can think of it as a single one. The 24 transcription factors together. Let's look at an entirely different experiment and see, and you try to come up with a way to isolate the variable of interest. The variable of interest is the one we want to know what causes in our experimental system. Okay, so again, the problem is to isolate the variable under study. Suppose that the study we want to do, and this means that we should maintain constant all variables, all other variables, except the variable under study, right? That's what we just. Let's suppose now that we wish to analyze whether the transcription factor E2F1 induces cell proliferation. This is a mistake. Transcription, not transcription. Okay. I'll show you. Actually, do you know how would you determine whether this transcription factor modifies cell proliferation? How would you approach this problem? You want to know whether this transcription factor accelerates or slows down proliferation. What kind of an experiment would you run? It's very simple.
Like the...
No, no.
No.
Women first,
Maybe we
especially shy women.
should.
They must be encouraged.
We should transform some cells and put them in a ground.
Medium, culture medium.
Yes. And with the wild type, so we have transformed the wild type with the same exact condition and medium, and we have to analyze during time how much the wild type and the transformed cells grow.
Good. Do you agree?
Yeah.
Okay. Anything to add? Just perfect.
Maybe a knockout gene.
Yeah, yes.
Sorry?
A knockout...
Yes, we might do a knockout,
Yeah.
right? But we'll choose the easier way. Doing a knockout is more difficult than just transducing the cells with the transcription factor. And let's say we have two initially identical dishes again, and we transduce one of them. In this case, we transfect the cells in one of them with a plasmid encoding E2F1. Right? And then, of course, in the beginning they have an identical number of cells in them. Then we count them, say, 48 hours after the transfection. And we find that the first dish contains 10 to the 6, 1 million cells. The second one contains 2 times 10 to the 6, 2 million cells. What do we conclude from this experiment? What do we conclude from this experiment?
The transcription factor doubles the speed.
It doesn't necessarily double the speed because we chose a single point in time. We don't know the dynamics of this, but it increases the speed of cell proliferation. Correct. Are we satisfied with this experiment? Have we, the question is, have we really isolated the variable of interest? And so the first question is, what is the variable of interest? What is the independent variable? Pretty easy. It's in the question.
E2F1.
E2F1 is the variable of interest. And so we want to isolate E2F1. Make sure that it is the only difference between the two dishes in the beginning of the experiment. Is it the only difference in this experiment, designed like this?
We didn't have the design vector.
Sorry, I didn't hear you.
If we do a transfection, there will be a vector.
Uh-huh.
So we should have an empty vector.
Exactly. So what Daniela is saying is that here we don't have only E2F1. We have E2F1 in a vector. How do we know that the effect we observe is not due to the vector rather than E2F1? We know it if we have a control, a proper control. A control in which we transfect only the vector, an empty vector is another cell. We might get even more sophisticated than this. At times people don't really remove E2F1 from the vector. They clone it in the opposite orientation so that the gene doesn't code for any protein anymore, but it's still there. The idea being perhaps there are sequences which do not code for a protein and yet affect cell proliferation in ways that we cannot predict on paper. So let's make sure that it's not the sequence, but it's really the protein encoded. So if I flip the gene, I still have the sequences, but no longer have the protein. But anyway, this is a very minor detail. This way the experiment is complete because we really have isolated the variable of interest. And let's say that we count cells here as well, and we find that they are 9 times 10 to the 5th, 900,000. What do we conclude now? The experiment is telling you something else besides what you're interested in. Experiments nearly always tell you more than you are interested in. Sometimes the bit more is more interesting than the thing you were interested in. So, if we leave the cells completely untreated, we have 1 million cells. If we add it to F1, we have 2 million cells. Clearly, this is due to E2F1 because the plasmid alone doesn't actually increase the number of cells. In fact, it decreases that slide. It decreases that number slide. So, what we conclude about this particular so-called experimental part, a single part of the experiment, what is transfection, plasmic transfection, doing to the cell? Compare this with this.
It's deleting some important gene.
No. Why are you postulating something like that?
Because
We only look, sorry, I'll let you finish in a second. We are only looking at the outcome of the experiment. We are not trying to make up theories. But anyway, finish what you were saying.
just because the number of cells was
Smaller.
smaller, so I thought that maybe some gene was
Deleted. Well, that's a really unlikely possibility, not inconceivable, but really unlikely. It's easier to perhaps to imagine that the plasmid is somehow poisonous, somehow
oh,
detrimental to the cells, or that the transfection technique in itself is detrimental to the cells, and that's what it is in fact. So basically the cells grow a little later, a little less, because they are impaired by transfection. But anyway, now the experiment is complete, because we have really isolated the variable of interest, and we are allowed to conclude that E2F1 does increase the proliferation speed of the cells. Now, this is, as I've said, isolating the variable of interest, the independent variable, is the privilege of experiments. And yet, the number of potential variables here is huge, immense, nearly infinite. So, how do you make sure that in fact every possible variable is identical in these three dishes? Isn't it possible that because you cannot handle them all together at the very same time, you're doing one at a time, you're working with one of them at a time, they are separated by a few seconds maybe, but perhaps having treating this dish before this one makes a difference. Perhaps this was, say, in one incubator, and you put these other two in another incubator. They have the same temperature, the same conditions, but they are not really the same. And there are many, many other potential variables. Perhaps this dish got contaminated. This is very frequent in tissue culture experiments. You have a bacterial contamination which is not visible even at the microscope because the bacteria are few, but they usually are dangerous or kill the cells, or anyway impair their ability to grow and to live, impair their health. So, while logically an experiment always gives you certainty of the results, because logically you have only one variable that distinguishes the experimental point in the beginning. In practice, there is an infinite possibility of unwanted variations among the tissue culture, the experimental points. Sometimes, well, first of all, so in doing experiments, being careful, being scrupulous, being attentive is never enough. Even if you're messy in your everyday life, when you're in the laboratory, when you're doing experiments, you have to be extremely careful, extremely organized, extremely thoughtful. Make sure that you're really treating your experimental points in the same way. And even with that attention, sometimes things escape your attention because you don't know about them. You cannot control variables that are influent on the results, but you don't know they are. And I'll show you one example. This is an experiment with mice. This is a typical so-called castle in which cages, here, plastic cages, transparent plastic cages, are arranged in order to put together the mice in a small space. So they are plastic and transparent because you can look at the mice without removing the cages and disturbing the animals. And let's say you are experimenting on a drug. You want to know whether this drug affects whatever you're interested in. Let's say, for example, anxiety. Surprisingly, anxiety, of course, is a psychological state. We know when we are anxious, but the animals cannot tell us. But we can deduce that the animal is anxious because he's shy, he tries to retreat, tries to defend himself, so that means he is anxious. There are parameters that you can measure and to determine whether the drug helps the mouse against anxiety or the opposite, or provokes anxiety, whatever you are interested in. So, let's design the experiments, and let's say that the mice in these two bottom rows of cages get no drug. These two other rows get, say, 10 milligrams of the drug per kilogram of body weight. That is usually the way you measure the amount of drug. Of course, a mouse doesn't weigh a kilogram, but still you do the calculation. Same as for humans. These two get 20 milligrams per kilogram of body weight. These two get 40 milligrams, and so on, up to the top. So increasing amount of drug. And then we measure anxiety parameters in all of these mice. And we find that indeed the drug is good because it eases anxiety. Excellent. But there is a problem. A problem you'll never guess, so I'm not trying to ask you what it might be. In fact, there are two problems, at least. The first problem is in the way I designed the experiment, the way in which I described it to you. We have the control animals at the bottom and the ones that take the drug up and down in the passage. That's not the way a good experiment gets done. Why? Well, for a variety of reasons. These mice get less light. These mice are probably slightly colder than this one because cold air goes down and hot air goes up. So the way a proper experiment with mice is done is by rotating the cages periodically so that every mouse occupies all possible positions. This is to eliminate positional effects. Also, one thing I didn't add to, not to make things too complicated, the control mice, the ones I've said would be at the bottom, but they wouldn't, do not really get nothing. They get the solvent of the drug. The drug is administered in liquid of some kind. They get the solvent because, again, we want to isolate the drug. The mice are identical under every respect except the amount of drug they've taken. So all of them must take the solvent. Good. But there is a variable that you will never think about. Look at this experimenter here. She's a woman. It's been fairly recently, just a few years ago, demonstrated that mice are okay with female experimenters, but are either feared or become aggressive against male experimenters. So, if you're measuring psychological traits such as anxiety, having a female or a male experimenter makes a difference. And perhaps you are shifting, you have thorns, of course. Not every time the same person goes to the animal house, and so if you do not control for the sex of the experimenter, you might introduce a bias that you are completely unaware of and draw the wrong conclusions. And so this example is here to illustrate that there is an infinite amount of variables that you will never think about. And so, although logically an experiment is conclusive, in practice you would rather run it twice, or 3 times, or more. You would like to have it repeated by somebody else. You would like to have it repeated, if possible, at all in another laboratory and so on. We'll talk about redundance of evidence about this in one future lesson. But remember these 2 things. In principle, an experiment is definitive. In practice, you should always be careful about the results and the signals. Let's go back to Jan. Let's go through at least some parts of his paper. Now he knows that presumably the 24 transcription factors can convert the cells. All of them, some of them, one of them. How would you find out? Is the question clear?
Can you repeat, please?
We know that if we put the 24 factors all together on the cells, we get something. But do we need really all 24 of them? Or is it just a few of them that do the trick and the others are simply irrelevant? Or perhaps it's just one among them and all the others are totally irrelevant. How do we distinguish between among the possibilities? And how do we find out which are the relevant ones, if any? If only some of them are. Yeah.
We start doing the same experiment, but when one transcription factor less every time.
What do you think?
When we change them.
Yes. Every time we eliminate one factor, and if one of them still works with 23 factors, of that one we're going to eliminate one more of each one, clearly, and so on, until we reach that one point that eliminating factors, it doesn't work anymore.
Okay. Remind me of your name, please.
Valeria.
Valeria. And you are?
Ilaria.
Ilaria.
You could also try one virus at a time and see if they work singularly.
Uh-huh.
and add them also in couples and
Okay.
with one.
Okay, we have two suggestions. You two say, let's eliminate them one by one at a time. She says, let's try different combinations. I prefer the first possibility. You know why? Because the possibilities, the combinations, are an immense number. So you might never find the right one. It is better to slowly eliminate the factors one by one and determine which ones are relevant and which ones not. So that's what Yamanaka did. He infected the cells with 24 transcription factors minus 1. Minus transcription factor number 1, minus number 2, minus number 3, etc. Now, this is a little complicated as a figure, but let's look only at the top, at the black top of the histograms. This is the number of colonies of G418 resistant colonies that you get 16 days after the infection. So, do the infection, you wait 1 or 2 days, then you have G418 to the dishes, and then you give time to G418 to kill all the non-converted cells, and you give time to the converted cells to grow and form colonies. Then you count the colonies. This is the experiment. Okay, so what transcription factors would you eliminate? I wonder. Just mention 1 or 2. Which ones would you eliminate? Oh, notice. I should say. This is mock infection. Let's see what it is for. And this is all 24 factors together.
Mm-hmm.
Okay. Now, what transcription factors would you remove from the pool?
14, 15, 20.
14, 15, 20. You would think that they are not needed to convert the cells. Do you agree, Ilaria? Do you agree, Daniela?
No.
No. Daniela does not agree.
No, because you don't have colonies when
Exactly.
you...
If you remove them, you don't get any colonies. So they are necessary to get the colonies. What you should remove is this, number 9, I think. Yes, 9.
Okay.
Right?
So,
Because without 9, you still get the colonies. Okay, so Yamanaka takes out 2, takes out 6, 7, 9, and it is left with 10 factors. And then he repeats the trick, removes them one by one, and eventually, it goes like this, and eventually finds that only 4 factors are necessary to convert the cells. And these are these ones: CMYC, KL4, SOX2, OCT3, 4. These are now well known as the Yamanaka factors. And again, if you transform cells, if you convert cells with the 4 factors here, you get cells that grow in a dish similar to embryo stem cells, natural embryo stem cells. If you use only 3 of those factors, yes, they do no longer grow like fibroblasts, but they don't grow like these. So you seem to need 4 factors. Okay? Now, let's focus on these 2 last experimental points: all factors and mock. We have already reasoned about what the mock is for. We have said, Daniela said, that it is necessary to make sure that the colonies don't form by chance. This is a so-called negative control. And negative control controls for the specificity of your experiment, which means the variable you measure at the end of the experiment is really due to the variable interest, to the independent variable, and nothing else. Here you don't have the independent variable that we have defined as the collection of the 23 transcription factors, and you don't get any columns. So that tells you that whatever you observe is specific to the variable of interest. What about this? What would you call this? All factors, all 24 factors together. What is it?
Positive.
It's a positive control. If this is a negative, this is a positive. A positive control is needed in the first place to make sure that the experiment is working. We had already seen in the previous experiment with all 24 factors that the 24 factors work. So now we have a condition that gives us colonies, for sure, we know that. And we use it as a positive control because there is a possibility, there's always a possibility in an experiment that you get all negatives. Suppose that all 24 factors were necessary to convert the cells. Then you will get 0 colonies all over the experiment, right? Because if they are all necessary and we remove them every time one of them, you will never get colonies. Now, this is a negative outcome. The result is negative. But in order to validate a negative result, you need a positive control. Because otherwise, you wouldn't distinguish a true negative result from a simple experimental failure, an accident that occurred that made your experiment fail, right? So, let's say all this in a more organized way. First of all experiments must include positive and negative controls. This is a must. You'll see in practice that in the laboratory that sometimes people don't do negative experiments or don't do positive experiments. This is not the proper way of designing an experiment, but sometimes you absolutely are totally confident about the fact that the experiment is working technically. So you do without the positive control. Or you are sure that you will not get negative results. So you don't use the negative control, sorry, positive control again. Or you know that the specificity is assured and you don't use the negative control. Okay, when you publish final results, you must have the controls. Sometimes to do the experiments in a faster way, in the routine of the laboratory, you might omit the some controls, but you should be aware that an experiment without the proper control is not a proper experiment. That is very important. And positive and negative controls have two purposes each, but they are not the mirror of each other. They are not the opposite of each other. So let's go systematically and consider. A positive control is necessary to make sure that the experiment is technically sound, that it gives the expected results. We already knew what were the results expected with 24 factors, so we use now the 24 factors as a positive control. The second purpose of the positive control is to validate negative results if they occur. Again, if we had all negative results here, we would need desperately a positive control to make sure that the experiment is working. So these are the two functions of the positive control. The negative control also has two functions. One is to verify the specificity of the experiment. Once again, here colonies arise if you have transcription factors. If you don't, they don't come out of the blue, they don't appear magically. So the experiment is specific. Also, a negative control is necessary to determine whether there is any background and to measure, to estimate the background. What is the background? In this experiment there is no background, and there are no colonies that arise spontaneously. But this is not always the case. Many experiments have a background. That is, the variable you are measuring at the end of the experiment, which is a dependent variable, because it depends on the independent one that you're isolated. The variable you are measuring at the end of the experiment is not zero in the negative control, it's more than zero, for a variety of reasons. There might be technical or biological. The technical reason is because your instrument doesn't give you a zero. It oscillates a bit, so you have so-called noise. You have values, it gives you values that are not zero, even in the absence of the signal you are looking for. Another possibility is that your biological system doesn't have a zero background. Suppose, for example, that 1 in a million cells spontaneously converts into an embryo stem cell. You would have one colony every here and there. And there would be a non-zero background. What would you do then? Then you would subtract that background from the results. If you got, say, 10 colonies here, then you would say that the result here is not 30, it's 20, because 10 is the background. So, the two purposes of the negative control is to determine the specificity of the experiment, and second, to estimate, to measure the background, and if necessary, subtract it from the results. Good. Now let's go back to Yamanaka's problem, which is interesting. Yamanaka wanted to make sure that what he got, the colonies that grew, were in fact embryo stem like cells. So, what would you do? You have a bunch of cells, and you suppose that they are like embryo cells. How would you compare those cells you have obtained here with real embryo stem cells? What would you measure, assess, observe? What would you look for? It's pretty easy.
The capability of
You can say it in Italian if...
differentiabile, differentiation capability.
Not bad, not bad. This is what Yamanaka will do later. But first, you have an easier way to preliminarily assess, because that's a complex experiment. It can be done, and it has been done, but it's a complex experiment. And Yamanaka wanted to know fast whether it was true, because it was a great result. Certainly, he was jumping up and down the lab and wanted to know the next morning if it was really true. What would you do? What determines the essence of a cell? What determines what a cell is? It's very trivial. What determines the fact that this is a fibroblast?
The transcriptome.
Exactly, gene expression.
Yes.
Whatever the cell is depends on its gene expression. We've said it when we talked about the Gardner experiment. We ruled out the possibility it was gene deletion, so it must be gene expression. All right, so you compare the transcriptions of the cells. At that time, it wasn't easy to do it like to do it today by NGS, next generation sequencing. You would sequence the whole transcriptome and look at all the genes together. At the time, that wasn't easy. So Yamanaka did a series of PCRs for a number of genes known to be expressed in natural embryo stem cells, and he wanted to know whether those genes were also expressed in these cells. So, here are the genes. Here are embryo stem cells. All of the genes Yamanaka looked for are expressed in embryo stem cells. They've been selected for being expressed in embryo stem cells, so of course they are expressed in embryo stem cells. And they've also been selected not to be expressed in mouse embryo fibroblasts. Otherwise, there wouldn't be a proof of anything if they were expressed in both naturally. All right, so ECAP1 doesn't matter. There's an explanation for why ECAP1 is not expressed in the Yamanaka cells, and so we don't care about it. Most of the others are expressed both in embryo stem cells and in Yamanaka cells. We should focus on these middle columns here, because these are induced IPS, induced pluripotent stem cells, obtained with 4 transcription factors. These have been obtained with 10 transcription factors, these have been obtained with 3 transcription factors, and we already know that they do not behave like natural stem cells. Okay, so you find that ESG1 is expressed, NANOG is expressed, ERAS is expressed, the last maybe 9. This is non-quantitative PCR, so the intensity of the bands really doesn't matter. It's a yes or no answer. GDF3, etc. SOX2 is not expressed, but there is a very valid explanation. SOX2 is among the 4 Yamanaka factors. So the endogenous SOX2 is not expressed because the exogenous SOX2 is already expressed, and the cell doesn't need to activate the endogenous. And all the others are expressed. All right? So at this level, really, these cells look pretty much like natural ES cells. But at this time, we are talking about positive and negative controls. The experiment is still the same. The variable of interest, the independent variable, is still the transcription factors. Let's say the 4 transcription factors here. But we are doing an experiment to, in fact, to be honest, this is an observation, not an experiment. We are taking the cells as they are. We are no longer manipulating them. We just look at them and determine whether they express the genes we are interested in. So if we do not manipulate them, and particularly if we do not add a variable, an isolated variable to any of them, this is not an experiment, it's an observation. We look at things as they are. But observations also need positive and negative control. So I'm asking you, where are the positive and negative controls in this experiment? Just a second. Let's give the shy girls time to...
Always on the right.
On the right is good. Specifically?
Yes, and left.
Okay. The ESL column is what? Positive or a negative
Positive control.
Positive control?
The other one is the negative.
Obviously. Okay. By the way, I'll draw your attention to NAT1, which is expressed both in ES cells and in MAPS. This is an internal control that's used in PCRs to make sure that PCR is working. Basically, it controls for the amount of RNA you've added in the PCR. These bands should be all similar to one another, to demonstrate that you have added the same amount of RNA in the PCRs. It's technical, we won't talk anymore about that. Okay, so correct. Positive control, negative control. Now, remember what's the purpose of the positive controls. The purpose is twofold. To show that the experiment is working, it is working, we have bands here. The variable we are looking at the end of the experiment, of course, of the observation, is a band. So, obviously the bands are appearing with their respective. And we don't have negative results except here, but they're expected here. So we don't really need the other function of the positive control that is validating negative results. On the other hand, the negative control here ensures that your PCR is specific. Cells that are not supposed to express the genes, in fact, do not give you bands. In PCR, it is common to have spurious bands, things that get amplified, let's say, by chance, without entering in the details. So you want to make sure that you are specific. This is very important. This control is very important in this kind of observation. And the other purpose of the negative control is to determine whether there is background, and if there is some, to measure it. But there is no background here, there are no grounds. So again, here, like before, we have a zero background. That is not always the case. Most often you have background. All right, good. Except we have another completely negative row here. It says RT minus, which means reverse transcriptase minus. Any idea what it is for?
Since we are looking at the gene expression, and we are not doing genomic PCR, we use a reverse transcriptase enzyme with who pairs to the polyadenylic tail on mRNA, and we amplify the gene to get cDNA. So
we actually retrotranscribe it into a cDNA, DNA copy of the RNA.
Yeah, I don't know what I say. However, the RT minus is for ensuring that you are not looking at genomic DNA.
Exactly. So this was not easy to guess. So obviously Daniele knew about that. You would not guess what it is for if you didn't know. Yes, this is correct. With when you are measuring or detecting, in this case, you are not measuring because this is not quantitative PCR. So you're not just detecting whether gene expression is there or not. When you are detecting gene expression, you are looking at RNA. So PCR amplifies DNA. You cannot amplify directly RNA. What you do is you first make a copy of the RNA into cDNA, copy DNA, using reverse transcriptase. Now, the possibility exists, and it's frequent, that your RNA preparation is contaminated by DNA from the very same cells from which you have extracted RNA. So you have to make sure what you're amplifying is actually cDNA and not genomic DNA. The way you do is this. You make sure that by not adding reverse transcriptase, you don't see anything. So this is again another negative control. Because I haven't said that positive controls and negative controls are unique. In fact, you might introduce several positive and several negative controls in your experiments, and we'll see examples of this, depending on the kind of experiment. So I would say that this meth is a biological, negative control. So he controls for the fact that these cells do not express those genes. Controls for biological reality. He controls for the fact that those cells do not have in them anything that might yield bands here. This is a technical control. Make sure that the way you have executed the experiment technically does not yield bands where they are not expected to be. So these negative controls ensure that the critical step of your observation, the isolation, the proper isolation of RNA, has been performed properly. Okay? Good. Let's go back for a second to the initial Yamanaka experiment. We have said that an experiment should always have a negative and a positive control. Now, we have already suspected that Yamanaka is a poor scientist, and in fact, the suspect comes up again, because this experiment has a negative control. I don't see any positive controls here. So Yamanaka was really sloppy designing this experiment, was he not? Before you answer, I'll tell you where a positive, how you devise a positive control. A positive control is whatever condition that gives you the kind of signal you are looking for. In this particular case, the signal you're looking for, the outcome you're looking for, is colony formation. So Yamanaka, to do a proper experiment, should have added another dish here in which he was sure that colonies would form. Right? What would be that positive control? This is a trap question. How would Yamanaka prepare a dish so that he was sure that there would be somatic stem cells converted into embryo stem cells? Valeria shakes her head. Because you don't know, or because, or for some other reason?
Maybe there is no way.
There is no way. Nobody was able to do that before Yamanaka. How could he possibly have a positive control here? He could have had a technical positive control. He could have engineered cells to express beta-GIO and make sure that they grew. It would have not been insignificant because one wants to make sure that the amount of G418 you add to the cells is not too much. Even if a cell is resistant, if you have too much G418, you eventually kill it. So that's really very minor quibble. In fact, it was impossible to have a positive control here. At least not a biologically significant positive control. He had a positive control right in the second experiment, because now he had a condition that could give rise to colonies. And so he had a proper control in the next experiment. Now, it turns out, Yamanaka told us 10 years later in an interview that in fact this was a crazy experiment, would have been a crazy experiment, except he didn't want to run an experiment here. These were preliminary experiments that he was doing to set up the system. But the system worked right away, even beyond his expectations. Sometimes experiments are better than you expect. It happens. And so he was really surprised that he got the thing to work right away. right, so let's continue. Positive and negative control. Let's say that we had an independent variable to, again, one of the usual two issues. Let's say this variable, we call this the independent variable x. We have the substance in the in the cell culture medium, which is now present here and not here. So it's an isolated and independent variable. We want to know what this substance here causes to the cells. Oops, and what do we do here? What do we add to the cells? Certainly not x, because we want x to be only here. What are we doing here? Remember, we want to isolate the variable of interest, which is x. But we are using a micropipette to put x into the cells. Micropipettes are used for doing what? To measure and manipulate what?
Fluids.
Fluids.
The medium. We put the medium without the...
Exactly, we put the solvent, like with the mice. We wanted to make sure the mice were affected by the drug, not the solvent. So we gave the solvent to the control mice. Here we add in the solvent to the control dish. Okay, let's suppose then that we are interested in three variables in the end of the experiment, whatever they are. You don't have to name them. Oftentimes we're interested in a single variable at the end of the experiment, but sometimes we're interested in multiple variables. So let's assume here we're interested in three of them, because we suspect that at least one of them might be affected by X. We are not sure which one. We are not sure even in what sense. Whether it will be increased, decreased, modified, whatever. Okay, these are, of course, the independent variables. Here. Sorry, the dependent variables. Meaning they depend on X by design, because the experiment was designed so that whatever we observe at the end of the experiment depends on X. And what we observe is that of the three variables we consider, B is increased at the end of the experiment. So our logically inescapable conclusion is that X somehow increases B. Now, it doesn't have to be a direct effect. X doesn't have to act directly on B. It could be very, very indirect. There might be a chain of many different effects between X and B. But the conclusion is that somehow, in some way, X affects B. All right. If this is the test experiment, what is this? What kind of control? Negative control. Okay. Are we missing something?
The positive control.
The positive control. Do we have one? Yes. Okay, so this is a negative control. What would be a positive control? Of course, you cannot tell me what to do, because we haven't named these variables. But in principle, what should it be? We've said it 2 minutes ago. A positive control is something that Ilaria nods, so let's listen to Ilaria.
Something that proves that B is increased by...
Exactly. It's a known condition. It's a condition known to increase B. So we call it Y. We know that Y increases actually A, B, and C because we wanted to have a variable that increased all of them, because in the beginning we weren't sure whether X would act on A, B, or C, or a combination of them. So ideally we would have a positive control in which all three variables are increased, so that we know that the fact that A and C are not affected is not due to a failure of the experiment, because in the proper conditions, A and C increase. So we haven't failed to detect the increase. The increase is simply not there. So the negative results on A and C are validated by the positive control. The positive result on B is confirmed in terms of specificity because in the absence of X, the negative control doesn't show an increase of B. You might think that perhaps just because you're waiting some time, B increases spontaneously in your cells, but it doesn't. And this is ensured by the negative control. So, designing proper positive and negative controls might look a bit difficult, but it is only initially. After a little while in the laboratory, you learn almost instinctively to add positive and negative controls, proper ones. It will become a routine. Just make sure that you are fully aware that experiments must have positive and negative controls. It might happen that even your seniors don't really care enough about positive and negative controls. Even scientists are not perfect. So you have to be aware that controls must be part of an experiment. All right, so let's examine one final case. Let's say that this time we want to evaluate not the effect of E2F1 on cell proliferation, but the E2F1 happens to be a transcription factor. We want to determine whether in our particular cells it is active or not as a transcription factor. Because not all transcription factors are active everywhere. It depends on the environment. They might need co-transcription factors. They might need other cofactors that are not present in all cells. And so even if you add transcription factors, you might not have transcription. So again, we design a simple experiment. We transfect the cells with the plasmid carrying E2F1. We transfect the cells at the same time with another plasmid. Oh, by the way, do you know how to determine whether E2F1 has transcriptional activity in cells? What kind of technique is used?
Real-time PCR.
Real-time PCR might be a way, yes, but that's not typical. Other ideas?
Chemiluminescence.
Chemiluminescence used for what?
If we use the E2F1 promoter with GFP, it
Yes.
will be brighter if there is.
Other ideas? Daniel has already named two possibilities. There might be others. For sure there are.
Like gene reporter.
Gene reporter, yes, that's what the second possibility Daniela was talking about. Reporter gene.
Two hybrid system.
The two hybrid system measures the ability of proteins to contact one another, to bind one another. It's different. But your suggestions are okay. We'll skip PCR, which would be a possibility if we knew a target of E2F. We might determine whether that particular gene target is affected by the presence of E2F. If its transcription increases, then we know that E2F is active. But let's say.
No, I was just thinking.
Go ahead. What were you thinking?
Che se noi abbiamo, allora italiano perché sennò, che per il sistema a due libri, sì, per vedere l'interazione tra due proteine. Se sappiamo, magari abbiamo una proteina che può interagire con questo fattore di trascrizione, ci può attivare un'altra cosa. No.
When in
No,
two hybrid, you would only know that the two proteins interact with each other.
Okay.
Whether they induce transcription or not is not part of the assay.
Okay.
You might modify the assay in a way that it shows you transcription, but it will be very complicated, so I wouldn't do that.
Possiamo usare una sonda RNA o a proteina.
To do what?
Are we trying to understand if the factor is expressed?
The factor we assume will be expressed because we are transfecting it into the cells. So the factor presumably is expressed. We can prove it that it is. Let's assume it is expressed.
Then I didn't understand the question.
Oh, okay. So the question was, I want to determine whether this transcription factor transcribes something in the cells. Maybe it's inactive in these particular cell types. I know that in general it works, but I don't know in these cells. Okay, so I take Daniele's second suggestion and co-transfect a reporter gene. A reporter which has luciferase, not GFP, but GFP would be the same. Luciferase under the control of an E2F binding site, a nucleotide sequence to which E2F binds. Okay, and then I have two dishes. In one of them, I transfect the vector, the vector expressing E2F. In the other, I don't. In both of them, I transfect the reporter gene. So, this is clearly negative control. I don't have the variable of interest, which is E2F1, right? Here, I do not have E2F1, so, and then I measure luciferase activity in both cells, in both experimental cells. Okay, obviously there is a major increase in luciferase activity. It's a logarithmic scale. So we have a million-fold increase in E2F activity, in luciferase activity, which implies that E2F is really transcribing luciferase very actively. Here, nonetheless, despite the absence of E2F1, we do have some luciferase activity. How come? Why? Women first. We have already discussed this possibility in principle, although we haven't encountered it yet.
Oh, it's like background of the control.
This is background. Our experiment does have a background. Background of about 20. On the logarithmic scale, this is 20 or 30. And so, in principle, we should subtract this background from this value. But this value is 10 million, so plus or minus 20 or 30 doesn't matter. All right, so, okay, we would be tempted to conclude that E2F is active in these cells. But we already know that we should be more careful than that. Also, there is a lot of space here, and there must be a reason. What are we missing in this experiment?
Positive control.
Positive control, good. How would you design a positive control? Again, a condition which certainly gives us a signal, luciferase activity. There are a couple of possibilities that come immediately to my mind, and there are certainly many more. Let's distinguish first. Do we want a technical or biological positive control? What would you prefer?
both.
Okay, yeah. Having both would be even better. Absolutely. But let's
yeah.
consider them one at a time. Otherwise we don't-
Biological.
Biological, okay. What is a condition in which E2F transcribes? A biological condition in which E2F transcribes. Remember the way we have formulated the question in the beginning. We want to know whether E2F works in these cells.
And so we take a stem cell.
Not necessarily stem cell. We take whatever other cell in which we already know that E2F works. Because I've said in general we know that E2F works. So
Okay.
we have plenty of opportunities. Right. Okay, this is a biological positive control. Now, Ilaria will tell us about the technical positive control. Let's go one step at a time. What would be a technical positive control? I'm not asking how you would devise it, how would you design it. I'm asking, what should it do?
We use it for,
If you prefer, you can speak it out.
lo usiamo per essere sicuri che stiamo guardando, prima l'abbiamo utilizzato per esempio per controllare che non stessimo guardando DNA genomico, se non sbaglio.
But that was a negative control. It was a technical negative.
Ah, ok, positive.
Positive, let's remember what a positive control is for. Positive control, besides validating negative results, this is not the case, we obviously have a positive result here, is needed to
Sì, sì.
make sure that the experiment is working. So, some help from Daniele perhaps?
Vedi cosa vedi che l'esperimento funziona o mettere la luciferina a valle di un promotore magari dell'actina, di un gene sicuramente espresso.
Yes, yes.
Sì.
Somehow make sure that these cells express luciferase.
Ma
Even if we have to add it
Ma-
physically.
non ce l'abbiamo già, cioè non è già quello senza vettore.
Well, this is our test. We now know that these cells express, but before doing the experiment we didn't know. Perhaps we would have all negatives here and we would need then a positive control. You designed the experiment before performing it, so you had a positive control just in case. But you're right, when you have a positive, you no longer need a positive control. Oops. Let's say that I'm satisfied with positive controls. In fact, I haven't even drawn them here, although there will be positive controls. But I'm more concerned about negative controls, which again are needed to ensure specificity and to measure background. We have already measured background. How do we ensure that our experiment is specific? That it really, that luciferase is expressed because of E2F1 and for no other reason.
L'abbiamo letto, dico. No, quando è partita la slide, l'anti-vector.
Well, say it anyway.
Anti-vector.
An empty vector, exactly. So, I was hoping that I had cancelled it fast enough. So, yes, you had an empty vector, similar to what we had done before. So we make sure that it's not the vector, it's not some sequences in the vector, it's not the transfection itself, it's just E2F. And then, I should add, we might add further additional negative controls, such as, for example, not having the reporter gene. So we put any combination of the other ingredients here, but because we don't have the reporter gene, we still have only background within the experimental error. This is essentially distinguishable from these others. Okay, so these are additional negative controls because we expect them to give us negative, except for some background. But the question is, in fact, the way I've drawn this graph here, this later part of the experiment tells us something else. Where is the background coming from? The cells or my instrument that is measuring luciferase? Remember, I'm measuring luciferase. I get some signal here, but I haven't transfected luciferase into these cells. Where is the signal coming from? Is that a technical background or a biological background?
Maybe it's the instrument that is reading, just keeps on reading something in the background.
Most likely it's an instrumental, a technical background, because there is no luciferase here, unless the cells, unless the very unlikely possibility that these cells express something that mimics luciferase, there is no luciferase at all. So whatever signal we measure must come from our instrument, not from a biological reality. And so we come to the negative biological control.
But isn't the last row luciferase a luciferase?
No, sorry, this is a misnomer. This is it's a confounding name. This is the luciferase vector. You're right. I apologize. This is the luciferase without the E2F binding site. So, if things were the way I mistakenly said, there is no luciferase, you would have been right. Instead, here we are dealing with biological negative control because we do have luciferase, but we are not in the biological conditions to allow luciferase to be transcribed at high levels. Nonetheless, we see that it is transcribed at low levels, which is very common with genes. They're always transcribed at least a little. Okay, but this gave us the opportunity of discussing both sides of the problem. Oh, we are at the end of the lesson, so I'll skip the rest, which is basically details, especially, I'll be very quick if you allow me to further minutes. This is a way to introduce technical controls. A positive control would be simply to add luciferase in the reaction mixture. If I add luciferase in the reaction mixture and then I measure it, I must read something. If I don't, my instrument is not working. A negative control would be an anti-luciferase vector. Would be an anti-luciferase vector. So this time really no luciferase at all in the experiment. So if I see any luciferase, it must be technical, it must be instrumental. Specifically, the way we read luciferase is by adding luciferin, by adding luciferin in the reaction mixture. So luciferin transforms luciferase and converts it into something that we can see as light. Suppose you have a crack, a small fissure in your instrument, you will see the light that comes from outside. That would be a technical background. And so finally, I'll draw your attention to, this is the end of the Yamanaka paper, we can skip it. But just to mention what you said at some point is what Yamanaka did. He put his cells into a developing embryo, into a developing blastula, and he labeled them with GFP, and he found that the whole embryo was GFP positive, indicating that the IPS he added to the embryo contributed to all tissues, so they were capable of developing in all directions, in all lineages, very quickly. Okay, I'll draw your attention to the final page in each, or almost each chapter in your textbook, which is called Punti Notevoli in Italian, and we call highlights here. These are the main messages that you should be carefully retain from the chapter. First of all, you should analyze a single variable at a time and keep rigorously constant all other experimental conditions. This is the real basis of all experiments. Some variables may be non-trivial or hidden. Remember the sex of the experimenter. Very, very non-obvious. No one experiment can be considered definitive for practical, not logical reasons. Negative and positive controls are crucial elements of every scientific experiment. Never forget this. The controls may refer to technical and or biological features of the experimental system. It depends on what you want to control for. A positive control assesses the functionality of the assay. Oh, these are the statements of the two functions of positive and negative control. The positive control assesses the functionality of the assays, that the assay is working, and validates negative results if you get negative results. And again, you don't know in the beginning, so you have to have a positive control in case you get negative results. On the other hand, a negative control assesses the specificity of the results and measures the background in quantitative experiments, if there is a